Right, hello everyone. Welcome to our fourth webinar of FAIRDM's Data and Model Management webinar series. I'm Natalie Stanford and I'm the FAIRDM Community Lead, and I'll be your host today again. And FAIRDM is, just to remind you all, if you're returning or if you're new, is an initiative to support researchers, students, trainers, funders and publishers. In fact, anyone interested in data and model management in better data management practices. And we offer software, advice, expertise, and training which make managing your data and models in life sciences easier. You can find out more about FAIRDM at our website at www.fair-dom.org, or you can follow us on Twitter at FAIRDM_EU. If you have any questions regarding FAIRDM, you can get in touch with me at community@fair-dom.org. So first, we've got a bit of news from FAIRDM again, and this is about our user meeting. So we'll be hosting our first user meeting as a satellite meeting of the International Conference on Systems Biology. And the meeting will be a full-day meeting in Barcelona on the 15th of September. Good data and model management practices improve the longevity and impact of your research. So if you've never heard about data and model management, or even if you're curious about it, or even an expert and keen to exchange ideas, our user meeting is the place for you. And we have quite a few different projects that are already using FAIRDM that will be represented there, and we'll be having lots of discussions proposed by you. Today we're here with our guest Neil Swainston, and he's from the Centre for Synthetic Biology of Fine and Specialty Chemicals at Manchester Centre for Biotechnology in the United Kingdom. So, Neil, are you there? Hello, Neil?
Hi, hi Natalie, how are you doing?
Hiya.
Good afternoon.
Good afternoon. How are you today?
Yeah, I'm good, thank you. Yourself?
Yep, good. Looking forward to hearing you talk. So Neil Swainston's a Senior Experimental Officer at the Manchester Institute of Biotechnology at the University of Manchester, and he has several years of industrial experience in proteomics bioinformatics development, and he started his research career 10 years ago, pioneering work into omics data analysis and management, genome-scale metabolic modeling, and more recently DNA design and optimization, and directed evolution of enzyme optimizations. He's going to be talking for about half an hour. If you have any questions during the session, you can ask them in the questions panel below. And at the end of the presentation, I'll unmute everyone and see if anyone has questions direct they want to ask Neil. The webinar is recorded and we'll make it available on our website in due course. So over to you, Neil.
Okay, thank you very much, Natalie, for the introduction and also for giving me the opportunity to speak with you today. Fortunately, it looks like the internet connections to Europe are still working, despite some of them other connections perhaps not quite so well. And so I'm very much looking forward to speaking to you today. So as Natalie said, I'm working in the Manchester Centre for Synthetic Biology of Fine and Specialty Chemicals. And my background's in informatics, so I'll discuss with you today some of the approaches that we use, primarily in the design side of the synthetic biology cycle that we use. And obviously a lot of this this discussion will have will be focused on the informatics side of things. So I'll just, if I can fast forward, yes I can. So I'll just take you through what I'll be speaking about today. I'll introduce the centre, the SynBiochem Centre. I'll give a brief overview of the approach that we are implementing here, metabolic engineering. And then I'll go kind of specifically into the data sides of things, specifically the challenges that we are facing and hopefully solving in putting together a design pipeline to enable metabolic engineering. And that will involve a number of factors. The first is in pathway selection, so actually deciding which enzymes we're going to use in our synthetic pathways. And I'll introduce an approach that we're using, quite a novel approach, is from the data management side, and that's the use of graph databases to store biochemical data that is of interest to us and how we can use that in selecting pathways. I'll also mention some of the algorithms and tools that we're putting together in designing synthetic genetic parts, and that's fundamentally how do we encode enzymes and DNA in such a way that we can express them in our host organism to hopefully produce the target chemicals that we're interested in. And then I'll mention some of the community approaches to storage of genetic designs, and also some of the nascent and very new and developing data formats that we're using in synthetic biology. These are community standards that are becoming more widely used and widely supported by software tools. And those specifically are SBOL and SBOL Visual. And at the end, I'll mention some further challenges, and some of those belong perhaps to the experimental side of work. Okay, so the SynBio Centre, as I mentioned, is grandly named there. It's a 10-year grant funded by the BBSRC and EPSRC, and we're about a year through it, maybe just a little over a year. It's one of about 6 centres in the UK, and our focus, as I mentioned, is the production of target chemicals. Now, we've got a very good group, a very interdisciplinary group that is co-located with me in the office here in Manchester. We're about 15 people, and it's what I particularly like about the group is that we've come together from all different backgrounds, but yet we are co-located and we work very closely together. And that for me is a very useful and appealing part of the centre. It's in contrast to a lot of other kind of interdisciplinary groups, they are quite often spread either between departments and quite often geographically unlocated. But the fact that I'm sat together on a daily basis with chemists and the molecular biologists who assemble our designs, the analytical chemists who apply mass spectrometry techniques to test our designs, and I can work together with myself being a computational biologist with more specialists from computer science and lab technicians. It's a really almost necessary team. Putting together such a team is kind of necessary that we can enable and solve these challenges that we do have. Now, taken from the website, this is the kind of spiel that we've put up there. We're attempting to provide tools, technology platforms, and SynBio know-how to drive academic approaches towards industrial exploitation. So we are in talks with industrial collaborators, and that's a key part of the centre. And we're hoping that we'll produce new products and methods that can be applied to drug development, agriculture, chemicals, and we've got some grants in based around new materials. So as I've said, the approach that we're looking at is metabolic engineering. And what we're trying to do is to produce cell factories that will allow us to produce target compounds of interest. Now, we know that cells, naturally, microorganisms, can produce, through their natural metabolism, a range of useful chemicals that we can use. An obvious one is fermentation that we use to produce, that we've used for thousands of years to produce alcohol. What the synthetic biology approach enables is allowing us to take the metabolism of a given host and use that in tandem with synthetic pathways that we can express within the host to take a particular feedstock to our particular target chemical. And the target chemicals, as I've said, may be biofuels, chemicals, or pharmaceuticals. Effectively, we're piggybacking on the back of natural metabolism, augmenting it with an engineered synthetic pathway to produce valuable chemicals that we require. Now, the approach that we use is kind of typical of synthetic biology. It's very hard to find a synthetic biology paper that doesn't mention the design, build, test cycle. Now, I'm very much in the design side, this left-hand column. Okay, so we work on various things, including enzyme designs, the modelling, and the design of synthetic pathways. And we've also got kind of larger scale modelling that we do at the host cell level. So this kind of area, the design side, is very much computational. It's the focus of what I'll be talking about today. But as I've said, I work obviously very closely in tandem with my colleagues who are involved in the build side of things, which is the assembly of DNA and the production of synthetic organisms. And then the next step in the cycle is the use of various approaches, but in our group here, primarily mass spectrometry and metabolomics, to analyze the results of our synthetic enzymes and our synthetic organisms. Are we actually producing the target chemical of interest? And as you can see, that we can do it at a number of levels. We can do it at the level of individual parts, which in our case typically enzymes, devices, which we look at our kind of collection of enzymes that we can plug together to produce synthetic pathways, and the level of systems, which is effectively taking the host metabolism and adding our synthetic pathways to it. And it's very important to point out that this thing is a cycle. Okay, so the approach we use is a cycle. Over time, it would be great if we could design pathways and design organisms such that they produce the target chemical first time at a particular yield that we're interested in. Now that, at the moment, is not feasible. So the approach we use is go through numerous cycles where we take the results of a first iteration and use that to feed back into a subsequent design cycle. And we can go around this circle numerous times until we hit production of our target chemical at a yield and a purity that we require. So this, obviously, as you can imagine, involves huge computational data challenges, not least the fact that we're working across a number of scales and a number of levels and a different kind of different points on that, different vertical points of that design, build and test cycle. So we produce a certain amount of data from the design side of things. The build side can produce a lot of data in terms of molecular biology, and then ultimately we may have a lot of heterologous data from the test side. So I've mentioned mass spectrometry data, but we can also have sequencing data and fluorescence data and high throughput screening data. And managing all that and managing the kind of the sample tracking throughout the entire system is and will be quite a challenge. Another thing that we've got to face is that we're kind of flying by the seat of our pants a little. There's not necessarily a defined pathway through that cycle. This is kind of part of the research that we're doing, is trying to optimize our way through that cycle. But at the moment, we're kind of finding our feet. This is a new field, it's a very exciting field, but we're using non-established methods, and we need to, provide agile solutions. It's effectively the analogy that I put there, which I've heard. It's like building an aeroplane at the same time as flying it. So we're very, the design side and the informatics side are very kind of reactive to the changing needs and the changing requirements of the experimentalists that we work with. Another issue that we have, which is becoming less of a problem due to the efforts of the community, is that we're wanting to use and extend existing standards, but in such a fast-moving field, the standards that we're using are continually in development and flux. Okay, so we're pioneering new methods that may not be at the moment supported by data standards. And that obviously is a challenge, but it's something that as a community we can work on in developing the standards to match the rapid rate of changes in the experimental side of things. But like I've said, it's a great job. It's tremendously exciting. We're working in a very collaborative field. I've mentioned myself, the group here is very interdisciplinary, and communities are developing both in the UK and further afield. And fortunately, we're part of those communities. Another factor as well is that academia and industry are interacting quite closely on this. So yeah, there are challenges, but we're working together with many groups to overcome these challenges. So I'll take you through a bit of the design pipeline. So in the top left-hand corner there, we have a target molecule that we may wish to produce. And we've got numerous steps and numerous software tools that will take us from this target molecule through ultimately to the synthetic DNA that we need to, well, first purchase, assemble, and express in our hosts to produce that target molecule. First piece of software is called RetroPath, and that is used to actually select the enzymes that we need to add to host metabolism to produce the chemical of interest. So in this very simple example here, imagine we're producing chemical D, or wanting to produce chemical D. The RetroPath software provides us with a number of candidate pathways. In this case, we've got enzymes 1, 2, and 3 that we can augment to the host metabolism to produce that particular target D. Now that pathway itself is kind of hypothetical. You know, we may have the enzyme sequences. These might be known enzymes, but we need to take those enzymes and generate synthetic parts that will allow us to express those enzymes. And there's a piece of software that I'll introduce to you called Parts Genie that will allow us to do so. That will produce our individual parts with each enzyme represented by a particular part, and then we can stitch these together in various ways to hopefully enact the above pathway and introduce the host metabolism and produce our target of interest. I'll mention briefly this approach of design of experiments. This allows us to generate combinatorial libraries in a sensible way. If we are expressing multiple enzymes in a pathway, there's many kind of combinations of how we can arrange these parts, and those combinations can increase to quite a large number very quickly. So what we're trying to do with the design of experiments approach is to limit those to a sensible number that we can express and we can screen. And then finally, we've got different pieces of software that can be used to design little bits of DNA that we use to assemble individual parts. So that's the theory, or rather that's the practice. We're working on developing each of these tools in the boxes here, and I'll take you through a couple of those throughout the course of our time together here. So when it comes to pathway selection, we're asking the question, how are synthetic pathway designed, and specifically which enzymes should we use? There's a multitude of enzymes that are available to us that we've taken from nature, and the challenge is, I guess, optimizing that selection, picking those enzymes such that we stand a good chance of making our target. Now, the problem that we have is that although a lot of this biochemical knowledge is available in a number of databases, it's quite disjointed at the moment. There's quite a number, firstly, there's a number of resources that hold this information, but perhaps more importantly, none of these are connected. Okay, so the way chemical data and biochemical data is organized at the moment is usually in kind of little silos. So there's many good resources for chemical knowledge, KEBI, KEG, HNDB, PubChem, and numerous other ones. But that's kind of not all, the data in that is not necessarily always connected to pathway data, which we find in other databases such as Reactor. Similarly, we have more high-focused enzymology data, and that's held in resources such as Brenda and SABI-ORK. But the result is of using each of these kind of tools separately is that integrating software, integrating queries, and even kind of boring things like mapping names and IDs can be quite challenging. And another thing is that performing simultaneous queries across these different resources is quite difficult. So the solution that we've come up with over recent months is to generate a single integrated knowledge base which will hold all of this biochemical knowledge in such a way that we can query across it. And do it in such a way that we generate a solution and a system that is flexible enough to change without changing requirements. And the approach that we've used is to use graph databases, specifically one built on Neo4j, which is quite a new technology, and so it's quite fun to play with, but it also gives us the flexibility and the extensibility that we require. So I can introduce you to SynBiochem-DB, which is, like I said, an integrated graph database which integrates chemical knowledge, pathway knowledge, enzymology data, and also knowledge of the organisms themselves from which those enzymes originated. So we also hold taxonomy data. And it's an integrated knowledge base. It centralizes and integrates the knowledge that we wish to query over, which reduces the kind of the data mapping problem across different applications and domains. And it allows us to query across that data as well. So we can do integrated queries across all of those resources. And ultimately, we may be able to use it also as something to hang our experimental data on as well. So we can use it at the initial stage of designing the pathways that we wish to put together, but we can also use it further down the line as a means of aiding our experimental data interpretation. And hopefully, we'll be able to answer complex queries across databases and domains. And I've given an example query there, which is kind of slightly fanciful and slightly kind of, well, maybe not ridiculous, but it's quite a complex query. And we're hoping over time we'll be able to answer things like this. So just answering what flavonoids can we produce in a particular host, E. coli, with a certain number of enzymes, using pathways that are balanced in terms of energy production and consumption. We may be able to add the values of the chemicals that we're producing, so we can look at high-value targets. And then we can ask kind of questions like, do we have metabolomics data for the intermediates? What are the enzyme sequences required? Etc., etc. Now, doing that with disjoint data resources, as we have currently, is quite difficult. So that's the reason that we put these things together. So if you look at graph databases, these are relatively new, I suppose. And if we compare them to the kind of more traditional, if you like, approach of using relational databases, we'll find a lot of advantages in the use of graph databases. So most of us, I guess, will be familiar with relational databases. They're very well used, quite ubiquitous, and they're stable. You know, we've been using them for years, and they do a particular job. But they do have the limitations. One of the limitations is the rigidity of the data itself. Okay, relational databases, as we know, they store highly structured data in tables, and I think the key point is that the structure of the data needs to be predetermined before we put the database together. So we require developers to strictly structure the data used in their applications. So to some extent, we need to get it right first time. And there's kind of technical issues related to this. We can see in this kind of very simple example below, if we want to map a particular person to a department, then use of relational database involve the introduction of many join tables. So if we want to connect Alice to the departments for future or A42, then that requires a separate kind of data in the middle, a join table that is at the technical level, and it doesn't actually hold any of the domain knowledge. This is kind of on the technical side, but nothing to do with the domain itself. And also at a practical level, if we have a structure such as I described before, which holds chemical data and pathway data, enzymology data, etc., that involves querying across all those things, involves a huge lot of joins, a huge number of joins, and that increases the operational costs enormously. So we use graph databases. Graph databases are flexible and extensible. We can come up with a data structure and tweak it over time. You know, these things aren't fixed. And it's partly because the relationships of first-class citizens in the model, we actually explicitly define the relations between concepts and between nodes. And these are organized by type and direction, and the relationships themselves may include attributes themselves. So in this case, if we're representing the same kind of information as Alice belonging to 3 departments, the structure of the database kind of maps what we would do conceptually if we were writing on a whiteboard. It's just a case of specifying our nodes, which in this case, the person, Alice, and the departments here. And then we can draw specified relationships between them. Now this provides a very clear model of the domain, and it allows us to be focused on the kind of queries we want to ask of the database rather than getting bogged down in the technical issues of how we normalize tables and how we represent this with join tables. And it makes the process of querying the data a whole lot more simple. Now we query graph databases through a Neo4j language called Cypher, and it's built on the basic concepts and clauses of SQL, and it allows us to both query and update the graph structure with, as it says here, concise statements. Now the advantage of this is it eliminates SQL statements that contain many joins, and means that we can focus very much on the intent of the query and not the technicalities of how we join data together. And the data itself, as you can see, is quite clean. So the example here of matching, of pulling out people's names who belong to a particular department, the IT department, can be made in quite a concise Cypher query relative to the equivalent SQL that contains a number of joins. So it's quite a simple way of both structuring data and querying data. Now, SynBiochem-DB, as I said, is made up from kind of warehousing data from a number of primary resources. The chemical data, we're very fond of KEBI. We pull our chemical data out of KEBI. We also use pathway data from KEG and MetaNet Ref. We have enzymology data, well, sequence data that we can pull out of UniProt, and we also have the kind of the tree of life that we can take from the NCBI taxonomy. And to put these together, we just put together a very simple kind of collection of scripts which access either web services or flat file downloads, and just go through the steps of downloading the primary data, parsing them, formatting them, and using that formatted integrated data to populate a Neo4j graph database. Now, this is a screen capture. I'll take you through a few of them. This here represents a little look at the data from a chemical perspective. So because we're using KEBI, which is an ontology of chemicals, we can start asking questions across that ontology. We can start asking questions, what flavonoids and what flavanones do we have in our data set, or what are available? So KEBI can, and indeed does, structure chemical data in such a manner that related compounds are grouped together. Now, this is in contrast to kind of KEG and PubChem that represents each chemical as an individual. Strength of KEBI is that it provides us with these relationships. So we have these ISA relationships. We've got relationships tell us about tautonomers and conjugate bases, and it allows us very easily to query across chemical space. What we've done is also linked that to reaction databases. So as well as having chemical databases, we can have green nodes here that represents chemical reactions that we know exist in nature. So we can see that this particular reaction here has a number of reactants. Okay, so we can specify the metabolites that make up individual chemical reactions. Similarly, we can see that these trees are quite complex. For a particular chemical here, L-DOPA, we can see that this is involved in a number of reactions, quite a number here, and this allows us to query over this space and this reaction space and look for particular pathways to produce our chemicals. We also have, as I said, the tree of life. So we've taken the NCBI technology, the NCBI taxonomy, and arrange that in such a way that that is arranged in the graph as well. So we've got the full tree from, in this case, we can look at any particular flavors of E. coli, particular individual strains of E. coli, but we can go backwards from E. coli up the tree of life, and we can start searching for enzymes just in a particular kind of branch of the tree of life. And so obviously the enzymes themselves that we specify are proteins. So we have protein data introduced from UniProt, which is in turn linked to the reactions which they catalyze, but also linked back to the organism in which they're expressed. So although it's not shown on this slide here, we can start writing queries from organism to enzyme, to reaction, to chemical. So we've got this data that was spread about previously is now integrated in one form that we can easily query over with Cypher. So this is a knowledge base that we can hopefully build lots of applications on. Neo4j itself comes with a REST interface that allows us to either use the native web interface that I just showed you there in the previous slides, or we can write simple Cypher queries over web services that we can use for a number of applications. So we can use them in pathway design, or as mentioned, we can use it in metabolic data analysis. So this work is ongoing. We're hoping to publish it soon, and over time we're hoping to expand the database according to our needs, and spread and expand the scope of the database out, which is something that we can do with Neo4j, which would be less easy to do with a relational database. So pathway representations. When we generate our pathways, we generate them in a particular standard. So we use the standard SBML. SBML is a long-established community standard for representing models, and it effectively allows us to mark up this particular pathway here in a well-used, mature XML standard. contains much of what we need. So it allows us to define chemicals and metabolites, enzymes, reactions. We can even specify kinetics and kinetic parameters in there, which allows us ultimately to do in silico metabolic modeling of potential pathways. And kind of crucially, we can annotate our pathways and annotate our SBML with publicly available database entries back to resources such as KEBI and UniProt. Now, what SBML doesn't hold is designs of synthetic constructs. So it will hold the pathway data, but not the detail on how we encode that in DNA. That's using a different standard SBOR, which I'll come on to later. Now, one of the previous FAIRDOM webinars of Brett Olivier explains SBML in a lot more detail, and if you are interested in that, I will encourage you to look back through the previous webinars and take a look at Brett's presentation. So we come on to parts design. Once we've got our pathway, we look at how we try to look at how we can express that, how we can encode that rather in DNA. And so the enzymes themselves need to be designed as individual parts. So a particular enzyme here that we perhaps have a sequence of, we need to translate that in such a way into something that we can drop into our host and express the particular enzyme that we're interested in. And this involves a number of optimizations. One of them is the specification of these assembly sequences on the end. These bits at the end are kind of like almost sticky ends of DNA that we use to stick individual parts together. We want to look at ribosome binding sites. That's represented by this glyph here. And we also want to look at coding sequences, which is represented by the arrow notation here. And so we want to optimize these three parts, these three internal structures simultaneously. And one of the things that we're looking at is optimizing the codon sequences. Now, if we look at this table here, we can see that there's a lot of redundancy in the genetic code. If we wanted to encode a serine in DNA, for example, there are a number of codons that we can use to represent serine. There's 4 there, there's another 2 here. And if we just assume that there's an average of 3 codons for each individual amino acid, it more or less averages out to that, then even a kind of small protein of 100 amino acids in length has something in the region of 5 times 10 to the 47 potential reverse translations. There's effectively that many DNA sequences that we can put together to encode that 100 amino acid enzyme. But we do know, we know the particular hosts, so we're wanting to express this perhaps in E. coli. We know that E. coli, for example, favors some codons over others. So what we attempt to do is, when we're optimizing our enzyme sequence, is to preferentially select codons that are preferentially used in that particular host. So we can see there's quite a spread of this, and this differs from organism to organism. So part of the goal is to ensure that we're using codons that effectively the host is happy with. Another thing that we're looking at optimizing simultaneously is the ribosome binding site. Now, the ribosome binding site, in kind of a quite hand-wavy manner, controls or has an effect over the expression level of the enzyme. So the strength of the ribosome binding site, and also what we call the translation initiation rate, it depends on a number of factors. One of the factors is the complementarity of what this is called a Chandal-Garneau sequence. The complementarity of that with the corresponding sequence of the ribosomal RNA. Another factor is the gap between the Chandal-Garneau sequence and the start codon that is used to initiate coding. And we also need to look at the secondary structure of the ribosome binding site. Okay, so this particular any DNA, but this particular segment of DNA can form secondary structure upon itself, and in many cases we want to minimize that secondary structure to ensure that we get good expression of our enzymes. So we've put together a piece of software that takes each of these factors into account and performs a multi-objective optimization, so we can simultaneously optimize both the RBS and the coding sequence. And that's a piece of software that's ongoing, that we're putting together at the moment, called Parts Genie. Now this, like many of the tools that we put together, it's put together with the intention that our design team and our experimentalists can use them. So although we do have myself and a couple of other computational people within the group, the kind of goal is that we want to produce tools that they can use so we can go on to kind of new and interesting things and not actually be providing bioinformatics services ourselves, but almost, you know, enabling the build team, the experimental team, to do that kind of work themselves. So PartScene itself, it's got quite a simple interface. It allows us to specify the host organism that we wish to express a particular protein in. We can specify one or more protein IDs, UniProt IDs of enzymes that we wish to express. There's various other factors, including the initiation rate, the translation initiation rate, effectively the strength of the RBS that we want to specify. And we can also exclude various sequences which may be problematic down the line when we try to assemble these things. So it's just a very simple interface, bit of a click, and then in the background we run this multi-objective optimization. So what we're attempting to do is go through a kind of iterative process where we iterate towards a good enough solution which considers what we've got here, the codon adaptation index. That's effectively how suitable the codons are to expressing that host of interest. We want to ensure that we're getting close to the desired translation initiation rate that the user provided. We want to make sure that the invalid sequences that we specified aren't present in the solution that we come up with. And we want to avoid other problematic sites within the design. So this, on the back end, on the server side, this is running a simulated annealing algorithm to iterate towards a solution that was good enough to allow us to design the parts that we require. And so the output of this are reusable genetic parts that we can use in a number of constructs. And we can put together libraries of parts as well. So as well as just doing a single enzyme, we can optimize multiple enzymes simultaneously. And that way we can generate larger combinatorial libraries that allow us to perhaps try a number of different homologous enzymes simultaneously. And once we've produced this, the next step is to integrate the results of this, the output from the algorithm and the software, with data management systems. And we also want to use, as I mentioned earlier, community standards where possible. So we use a system, a third-party system for storage of genetic designs, and that's called ICE. This is the Joint Bioenergy Institute Inventory of Composable Elements, quite a grand name. But this is a very good piece of kit that's been put together by the JB Lab in Berkeley. It's a really fantastic web repository for genetic designs. And it's open source and it's extensible. Provides us with both browser and web service access, and it contains a number of tools that my colleagues in the build side can use in the annotation, manipulation, and analysis of the sequences that we put together. It's got a number of features that are kind of useful. It allows us to text search the metadata that we assign to our designs. It also allows blast searching of sequences. So if we have a particular sequence that we want to encode, we could initially do a first blast search to see if we already have that sequence or something similar already in the database. And it supports numerous data formats, and it's very carefully controlled in terms of user permissions as well. It's built on the idea of a web of registries, so it supports the idea of a distributed collaborative use. So we can choose to mix and match between both public and private registries. There's a publicly available online registry that contains a number of kind of standard parts that we can use in kind of simultaneously. We can use at the same time with a kind of more public repository that contains our own private designs. The publicly available version is available there. I believe anybody can sign up. You can have a bit of a play with it. And I'd encourage you to do so because I think it's great. I've had nothing to do with the development of it so far, but we've been very impressed with it. And the writers and the designers of the software have helped us out numerous times in putting this together. I'm sorry, can you see this little thing on the side of my screen here? I'm trying to get rid of that. Okay, there's also related pieces of kit that do the same kind of thing. These are other means of storage of DNA designs. There's the IGEM registry from the MIT, and also the SBOL stack from our friends and colleagues in Newcastle. Excuse me. So a few just screen captures. This is ICE itself, its web interface. It allows us to do kind of simple searches on expression vectors. We can, find, or whatever, text-based searches, find what's in our system. We can organize them into particular repositories and collections. We can look in details at individual designs, assign metadata to them, and actually have a graphical representation of a particular design. There's also scope for analysis tools on the right-hand side. We can link this to sample data and experimental sequencing data. And we've got this kind of permission thing, so we can limit who can actually see individual designs and also make them public as and when pieces of work get published. We also get this nice kind of plasmid view as well, and this thing in the bottom allows us to manipulate the data, the sequence data, and view it and look for features on the sequences. And then crucially as well, we can export the data in a number of formats. I'll take you through some of the formats now. The kind of more simple one that we can export data in, this is I guess at this point we're going into means of representing, kind of more standard means of representing sequence data. And the more simple one is FASTA, which I guess most of you will be aware of. This allows us to represent both protein and nucleotide data, and it's a very simple format that involves just the sequences themselves and a kind of almost like unstructured naming field. Next up, the kind of sophistication tree, I suppose, is the GenBank format. This allows us to specify both kind of metadata about a particular sequence, the sequence itself, but also allows us to specify features that are on that sequence: promoters, terminators, protein binding sites, etc., etc. Now that's a step up from FASTA, because it allows us to actually, add some flesh to the bones and actually specify the function, if you like, or the structure of our designs. But it's still quite problematic, still quite an esoteric, non-standard means of representing data. I think GenBank itself dates to something like 1982, and it almost like predates standards such as XML. And as such, it's quite hard to enforce compliance. There's no kind of equivalent of an XSD file or an XML schema that allows us to ensure that GenBank data is reliably encoded. So, based on this, the synthetic biology community, maybe about 5 or 6 years ago, started putting together a brand new standard for representing synthetic biology designs, specifically to allow the exchange of designs. And the focus of this standard, SBOL, was on machine readability. Because it's been produced rather recently, it's using kind of modern tools, Model Kit. So SBOL itself is written in XML, and it's specifically RDF based, so it's a RDF representation, an XML representation of RDF. Obviously, using XML and using RDF has numerous advantages. We can use many collections of XML processing tools that exist already. We get all that for free. But also we can structure our annotations a lot more carefully. Now, SBOL itself uses a specific ontology, sequence ontology, to allow us to encode those features such as promoter or coding sequence in a controlled and machine-readable manner. Now, SBOL itself is community-driven. It's in constant development, and as such, it's flexible and it's extensible to, as I say, these fast-moving requirements that we have in synthetic biology. And it's designed to support the definition and reuse of genetic parts. And crucially, it supports hierarchical designs. So in the same way that you can do so with electronic circuits, a particular kind of plasmid or a particular synthetic system that we put together may be made up of multiple parts. So the kind of parts that I designed earlier that I showed you in parts genie, they can exist as reusable SBOL documents, if you like, and those can be integrated together to produce a full design. So it's effectively allowing larger sequences to be built up from subsequences, so it's very modular. And it also supports iterative design, so we can specify things on an ongoing manner. So we may know that we want a chunk of DNA of, or a ribosome binding site of length 60 nucleotides, but we might not know specifically what that sequence is. So it might be, maybe the tool of a piece of software to produce a ribosome binding site of 60 nucleotides. Now, we can't represent that information in GenBank, but we can do so in SBOL. So it supports this kind of iterative design process that we're doing. There's a little glimpse of SBOL there. Obviously looking at XML isn't necessarily the nicest thing to do, but you can see the flavour of what we've got here. This is actually SBOL 1. It allows us to specify certain metadata and certain names about sequences. We've got the full level sequence itself at the top here. Look at my arrow back. But we can also add these sequence annotations that allow us to annotate particular subsequences within the top level sequence, where the subsequence starts and ends, which strand it's on. And we can also specify these sequence ontology terms, so we can annotate the sequence itself. Another thing that's worth mentioning is the use of SBOL visuals. So at the same time as producing, or kind of in parallel to producing SBOL, which is largely machine readable, it's for software interchange, the community put together SBOL Visual, which is a means of standardizing synthetic biology designs for us lot, for people, for humans. And SBOL Visual is designed to be analogous to the IEEE standards for electronic components and circuits, and as it says on the website, it's inspired by the clarity, simplicity, and usefulness of electronic symbols. So the whole idea, the whole goal of this is that in years to come, a synthetic biologist would look at that design there, just in the same way that a electronic engineer would look at a wiring diagram and know exactly what that means. So it's the kind of standardizing the representation of genetic designs. And at the moment they have glyphs for common sequence features, and each of these features are mapped to the corresponding sequence and ontology terms, and that allows us to map directly from the computational readable SBOL to the human readable SBOL visualization. The glyphs themselves look at functional components such as coding sequences or terminators, but also assembly methods that we use for stitching DNA together. And it's been designed in collaboration, or kind of in parallel with SBGN, which is the Systems Biology Graphical Notation. So that involves a mapping between the kind of the pathway data, the biochemical knowledge, and the actual DNA designs that we can use to encode those pathways. Now, as I said, it's a fast-moving field, so we've moved, the community has moved from FASTA originally through GenBank, through SBOL 1.1, which I showed you there, to a recent addition, which is SBOL 2, which was published and made available last year. And as well as supporting the structural information, as SBOL 1.1 did, it also specifies, allows the specification of individual modules with defined inputs and outputs. And there's another level of information in SBOL 2 which wasn't present in SBOL 1.1, and that's a focus on the functions and interactions between particular parts. So in SBOL 1.1, effectively it represented the DNA and the structure of the DNA, but it didn't really tell us a lot about what that DNA was meant to do. So some of the things that we may wish to represent more fully can be seen in this diagram here. So we may want to look at specifying, as well as DNA, the proteins that they encode, and also inhibitions and activation relationships. And this is now available in SBOL 2. It's a lot richer than the original SBOL 1.1 version. As an aside as well, it's also compatible with mathematical models, so there's a link between the kind of the DNA world here and also the mathematical modeling world of SBML. And work is ongoing and indeed has been published showing how one can interact with both SBML and SBOL simultaneously. Now time's moving on, so I'll have to wrap this up, I guess, kind of shortly. But just to give you an indication of the number of tools that are currently supporting SBOL, this is taken from the SBOL website, and currently there's about 25 tools and growing. And these are covering various different aspects of DNA design and analysis, and they run on various platforms, and they've got various availabilities. And it's very much an ongoing effort. We've got a number of tools that we're developing that are not currently on this matrix, and there's a number of groups also working in the support of SBOL, and I think that's going to grow over time. Just one kind of quick example, SBOL itself, as you've seen, it's XML based, it's not really made for biologists to interact with directly, but rather through some of the software that I was talking about. And I think the most simple example of a piece of software that supports SBOL is the SBOL Designer, which is freely available. And this is just a really simple kind of drag and drop interface that allows us to put together very simple designs, such as that one there. If you are writing software, such as myself, there are a number of libraries that can aid you in manipulating and reading and writing SBOL documents, and they're kind of different flavors of libSBOL. There's a site there, GitHub site, this is all freely available, and it supports both the original SBOL 1 standard and SBOL 2, and there's a number of programming languages that are supported. I personally use the Python one, but there's similar libraries available in C and C++, Java, and JavaScript. I won't mention the design of experiments due to time constraints, but what I will mention is this, given the relevance to what we're saying about SBOL. A really recent development in the last month is the recommendation by a major synthetic biology journal for authors and for submitters of manuscripts to make their data available in both SBOL and SBOL Visual. As it says there, synthetic biology, SES Synthetic Biology recommends that all authors digitally deposit their genetic designs using the SBOL data format, and also recommends that within the manuscripts themselves, authors visually depict the designs in the SBOL Visual standard. Now, this is a really good development because obviously we've had the problem in the past that designs have been present in papers in a kind of, if they're present at all, if the DNA sequences to support the particular manuscript are available at all, they're in a kind of, esoteric, non-standard, random format. And this, although it's a recommendation, it's a really good step in the direction of data storage and data standardization. So this is just one tranche of the work that we're doing. So this is just the design side, is what I've been talking about here. Obviously, there's another, maybe 3 webinars that we could do focusing on the amount of data that is produced both in the build and in the test side, but that, I'm afraid, we'll have to wait for another day. So this is just an indication with some rather nice pictures that we've got from our resident artist of the complexity of what we do, both in the build side and in the test side. So we've got additional data from mass spectrometry and from the lab that ultimately we'll need to manage and link to the designs that I showed. So in conclusion, that's where we're at. Okay, this is ongoing work. We've done an awful lot of work in the design side of things at the moment. We're looking to, as the experimental side moves on, we're looking to do more work on the data management of experimental data. And crucially, we've got quite a bit of steps to kind of link these designs through the multiple steps of that design, build, test cycle in such a way that we can kind of sample track from, what aspects of this particular design provided us with highly performing strains of synthetic organisms that produce the target that we're interested in. So, with that, I'll thank you for your patience and your time. I'll acknowledge my lovely team there. So that's the interdisciplinary team that I was discussing. Also, obviously, like to thank our funders, the EPSRC and BBSRC. And I'll leave you with some references that you can pick up and read more details on some of the tools that I was discussing. But finally, just say thanks. And if we've got time for any questions, then I'll be happy to take them.
Thanks, Neil. That was a very good presentation and very concise about what's been happening. So if anyone's got any questions, we'll take a couple of them. Obviously, if you have to go, you have to go. So if you have any questions, you can ask in the question panel on your control panel, or you can raise your hand and I'll come to you. I'll also may unmute people in a bit, but whilst I'm waiting for questions to come in, I'll kick off with the first one, I think. So what I noticed was how many different tools you're actually using within the pipeline just for that, one aspect of what you're doing in synthetic biology. How connected are these tools and how easy are they to use together in this pipeline?
Well, at the moment, we're at a kind of a non-enterprise edition state, if you like. So each of these tools are kind of freestanding at the moment, but I don't know if it came across necessarily as in the talk. Because we are dealing with standards, we're kind of hoping that the output of one of these tools is the input to the next one. Okay, so if we look at the first initial step of designing pathways using tools such as RetroPath, the idea is that what is produced from that is an SBOL document that is annotated in such a way it tells us exactly the enzymes and the sequence, well, not necessarily the sequences, but say like with Unicode terms, we can annotate them. So we know exactly the sequences of what we're wanting to produce in the next step, which is the parts genie side of things. And so by ensuring that the inputs and outputs of each of those tools, maybe not enforcing that they use these standards, but allowing them to read these standards, then we can have this kind of, at least initially, we can have this pipeline going through the, our designs. The individual designs of enzymes will drop into the ICE database, and we'll use them subsequently in subsequent steps to kind of stitch them together into pathways. So over time, we'd like it to be a little more kind of turnkey, I suppose. But at the moment, these will all play well, once we get these to completion, the intention is that they will all kind of play nicely together because of the use of standards.
Okay, so you see it's something that will develop as the field matures a bit.
Yeah, I think so. The kind of a lot of the motivation for what we've done so far is not reinventing wheels. Although we've got a good team here, there are only 2 developers, effectively 2 or 3 developers, perhaps, working on the design side of things. And so we don't want to just develop software for the sake of it. We're very much responsive to the needs of the group here. So we'll produce things when we need them. And if there's something else that already exists that we can use, then we recommend using that. The reason we put together things like Parts Genie and such like is because at the time we were looking, there wasn't anything that did this kind of multi-objective optimization that we needed.
Mm-hmm.
And so we kind of we wrote our own, but at the same time as writing our own, we reused kind of algorithms that currently exist. We maybe just pinch different bits of algorithms and put them together. So the kind of the RBS calculator that we use, the ribosome binding site, that optimizes effectively the strength of the expression levels of the enzymes. That was just taken from a GitHub site. The person who made that, a guy called Howard Salis in Penn State, very kindly made it freely available, so we could just use that software at the back end, use that code on the back end of our algorithm as just one of the things that we're attempting to optimize.
Okay, that sounds good. We have a question from Wolfgang Müller, and the question is, why Neo4j and not RDF? And I think that's with respect to the SynBiochem database.
Yeah, that's actually a very good question. I think Neo4j, one of the advantages of it, from my point of view, is that it was just new. There was it's a novel piece of technology. I was taken in by their description of how easy it was to use and how easy it was to query, and there's not necessarily any technical reason of using it beyond using RDF, apart from the simplicity. It really is to put together a database such as that, as I was describing, downloading flat files, parsing them and structuring them in such a way that I can populate a Neo4j database. It's a tiny amount of code, so there's very little overhead to actually populating the Neo4j database. Now that might be the same for an RDF triple store. I don't necessarily know. But the fact that this is available, it's easy to use, we get the nice web interface for free, and we also get the nice web service interface for free. It means that we can concentrate basically on the content of what we're putting in there, rather than the kind of technical means of how we're getting information out. So Cypher's quite easy to learn, and the database itself is quite easy to populate.
Okay. Sounds like a reasonable explanation. So I think, given the time, we're probably going to have to call it a day there. But thanks very much for the interesting talk. And if anyone has any more questions, you can either get in touch with us at Fairham and we can pass them on to Neil, or you could get in touch with Neil directly. I'm sure you can find his details on the SynBiochem website. With respect to the Fairham user meeting, if you go to the handouts, you can actually download a flyer. It's called the Barcelona flyer, so if you're interested in attending, you can get a bit more information from the flyer below. In the meantime, thanks for joining us again this week, and we hope to see you next month for our final webinar of the series. So thanks again, Neil. It was good to have you on our webinar this time.
Thank you very much. Really enjoyed it, and thanks to everyone for tuning in.
All right, thanks. Bye-bye.